Okay, it looks like people are filtering back in. We can go on to another one of these. Hopefully that's something you're enjoying. So here we go. So again, we're going to begin with from the very top with the key to California plant families, the key to groups within that. So specimens available for examination without flowers, these either not present on the specimen or not produced at all, or specimens available for examination with flowers, often also producing seeds within fruits, which is angiosperms. So once you get to know this key, all of these couplets become very, very familiar and you'll be able to get through them really quickly. Specimens with only cleistogamous flowers, fruits developing without flowers opening, or specimens with open flowers. So in this case, we have open flowers. Specimens with unisexual flowers of only one kind, staminate or pistillate, but not both. Plants dicecious or monoecious, or specimens with bisexual flowers or with both staminate and pistillate flowers on same or different individuals, plants bisexual, dioecious, or monoecious, occasionally with mixture of bisexual and unisexual flowers. So in this case, we do have bisexual flowers with both stamens and a pistil. Pistils 2 or more per flower, carpels more than one, free to the base, or pistil 1 per flower, carpel 1 or carpels more than one, fused at least proximally. So in this case, it does look like we have one ovary here. So this is the pistil again with the ovary, the style, and the stigma. Perianth 0 or in a single whorl, appearing to be either sepals or petals, but not both, sometimes reduced to scale-like or bristle-like structures. Perianth in 2 or more whorls, generally both sepals and petals, or perianth parts spiraled 2 or more times around floral axis. So in this case, we do have what are very clear examples of classic sepals and petals. So typical what you would expect for those structures. Perianth parts 2 or 3 per whorl, or perianth parts generally 4 or 5 per whorl, rarely some other number of whorls differing in number of parts, or in a spiral with the number of parts indefinite. So again, we kind of have to look from multiple angles, but we have 1, 2, 3, 4, 5 sepals, 5 petals, and 5 stamens. So we'll go with the second choice. Petals fused into a ring or a tube, the corolla generally falling as a unit. Corolla of free petals fused to a hypanthium should be keyed under 19 prime, or petals free, at least at base, attached and generally falling singly, sometimes individually joined to a hypanthium, and a few families more or less joined and falling in groups, but not forming a ring or a tube or petal 1. So in this case, these petals may look free, but once you remove the calyx, you can see that they really are fused at the base. So when they fall or come away from the flower, they're probably going to come as one unit because they are fused into a ring. So we will go with the first choice. Ovary inferior or partly so, or ovary superior. So again, always a good practice to pull away the corolla and have a good look. And this is a classic superior ovary. Corolla bilateral or corolla radial. So in this case, remember, this has to do with lines of symmetry that you may be able to draw through the flower. And in this case, it's radially symmetrical, so you could draw multiple lines of symmetry. So this will bring us to group 19, Perianth in 2 whorls, petals 4 or more, all fused, corolla radial, pistil 1, ovary superior. So that sounds like that agrees with everything we've just seen in our plant. So again, always a good moment to double check that that description matches your plant. It's really easy in a key to just get mixed up and go to the wrong line, even when you have made the correct choice just by getting mixed up. So this is always a good place to double check. Stamens greater in number than corolla lobes, or stamens equal in number to corolla lobes, or fewer. So in this case, we have 5 of each, so they are equal in number. Fertile stamens fewer than corolla lobes, 1 or more sterile stamens sometimes present, or fertile stamens equal in number to corolla lobes. So again, we have 5 of each, so they are equal in number. Plants entirely non-green, parasitic, or plants green and photosynthetic. So this is a rare condition to have non-green parasitic plants. In this case, we do have very nice green leaves, so we can assume that this is a photosynthetic plant. Ovaries 2, more or less pressed together in flower, but not fused. Carpels distally united by a shared style or by a complex stigmatic structure. Fruits of 1 to 2 follicles, milky latex generally present, is Opistinaceae. Or ovary 1, sometimes deeply lobed, fruits various, milky latex none. So if this had milky latex, you would expect to see it right here where part of the tissue has been removed and the vasculature has been exposed. And again, it looks like we have 1 single ovary, and this is really a clear description of Opistinaceae, which if you're familiar with milkweeds and their relatives, you will recognize this as not one of them. So we'll go with the second choice here. Leaves whorled, or leaves alternate, opposite, or all basal. So in this case, it's really hard to tell if the leaves are opposite or alternate when they're all clustered together. So the term basal is used when basically all the leaves are just coming off very tightly from the base. So the arrangement is not so important as to know that they are all clustered very, very closely together, basically on top of each other. So in this case, we have basal leaves. Corolla dry, scarious, 4-lobed. Inflorescence a dense spike, rarely flower only 1. Leaves generally all basal. Opposite, appearing clustered at nodes in Plantago arenaria, which would bring us to Plantagenaceae. Or corolla with normal petal-like texture, 4 to 7 lobed. Inflorescence various or flower solitary, leaves basal and or colline. So in this case, we do have very much petal-like texture, and 5 lobes fall in that 4 to 7 range. They're not 4-lobed. So we'll go with the second choice. Ovary deeply 2 to 4 lobed, especially in fruit. Style attachment surrounded by lobes of ovary. Style readily detaches in Lamiaceae as corolla falls. Fruit breaking apart into 1-seeded nutlets or segments, sometimes only 1 to 3 nutlets maturing. Or ovary entire or shallowly lobed, style clearly attached to tip of ovary, fruit generally a capsule, berry, drupe, or a keene. So this is a really nice example. You can see an entire ovary, and the style is really attached at the very top, and there doesn't appear to be any lobing. Filaments fused, forming a tube around style, or filaments generally free. So this is a great example of what Bruce was talking about with the fusion of parts. So we do have the filaments, which are often, filaments are normally very hair-like and thin, or very typically hair-like and thin. In this case, they are really broad and flattened and are forming this tubular structure. So this has been sort of torn, the flower has been sort of torn open. You can see the stamens and their fused filaments. So they are, we'll go with the first choice where the filaments are fused. So then we have a shrub, flowers solitary, yellow or orange to more or less red, stems and leaves densely stellate, corolla-like whorl, actually a calyx, subtended by sepal-like bracts, which would bring us to Fremontodendron in the Malvaceae, or herb, flowers in scapose umbels, white to pink or purple, stems and leaves glabrous or glandular hairy. So we know this is an herb, not a shrub. And so this scapose umbels is referring to this long peduncle is a scape. And then this is an umbel because each of these flowers are radiating from a single point. So it's a scapose umbel. So we'll go with that second one there. So this brings us to the family key to Primulaceae. So I'll just take a moment. If you are following along in the Jepson manual, this is one of those groups that has changed recently. So there is going to be an extra couplet that's going to come in. The generic circumscription has changed, but most of the parts of the key will be the same. There's just a new couplet that has been added in. So some of these are going to be renumbered. The corolla lobes are reflexed, anthers more or less adherent into a cone, leaves 1 to 60 centimeters, or corolla lobes spreading or erect, anthers free, leaves 0.3 to 3.5 centimeters. So in this case, we have very strongly reflexed corolla lobes, and the anthers are adherent into a cone. So this brings us to the genus Primula. And as I mentioned, this is one of the groups that has undergone a change recently. And so in the last Jepson manual, these were treated in Dodecatheon. So Dodecatheon turned out to be highly nested within Primula, and expanding Primula for the moment seems like the best taxonomic solution. So this is now treated in Primula. And then another thing that's really, really handy with reading these descriptions is that sometimes there's specific terminology to a certain group. So anther connective is really an important term when you're looking at this structure that fuses all of the anthers together. And that's kind of an unusual structure. So sometimes those unique words or terminology will be used in a specific key, and it'll almost always be defined within the description of that clade or that group. So it's always another good reason to read the descriptions. So key to Primula, corolla lobes spreading or erect, rounded, anthers free, leaf 1.5 to 3.5 centimeters, heterostylus is Primula suffrutescens, or corolla lobes reflexed, generally acute, anthers adherent into a cone, leaf 12 to 30 centimeters, homostylus. So in this case, we have the very reflexed lobes of the corolla and the anthers fused into a cone. So we will go with the second choice. Stigma enlarged, much larger than the style in width. Anther connective wrinkled, dark maroon to black, filaments less than 1.5 millimeters. Free or joined by thin membrane, but not fused into tube. Or stigma more or less not enlarged, more or less equal to the style in width. Anther connective smooth or wrinkled, yellow or dark, filaments 1 to 4 millimeters, fused into tube, or less than 1.5 millimeters free. So in this case, this is really important to remember the stigma. So that's the very tip of the style, which is the receptive surface. And we can see in this case, it is pretty much exactly the same width as the rest of the style. So that's a really good indication because some of these other characters are variable. So that's a really handy one. And then we do have these really dark anther connectives also. The anther connectives smooth to longitudinally wrinkled when dry, or fruit circumscissile or with valves, or anther connectives transversely wrinkled, fruit circumscissile. So this is really a key word here, when dry. So in this case, we're looking at fresh material. So we do have these really big, broad, transverse wrinkles. So knowing that this isn't dry and we do have these major wrinkles, we will go with the second choice. Leaf blade generally tapered to petiole, length generally greater than 2.5 times width, filament tube greater than 2 millimeters, connective dark, smooth, or obscurely wrinkled. If leaf narrowed abruptly to petiole, then filament tube less than 1.5 millimeters, connective dark, generally smooth, or leaf blade generally narrowed abruptly to petiole, length generally less than 2.5 times width, filament tube 1 to 4 millimeters, connective dark, generally wrinkled. So this is a challenging break, and it's also challenging to determine exactly where does the blade transition into the petiole. So that's challenging for getting a measurement. So one of the things that is often really helpful is that floral morphology tends to be more stable than vegetative morphology. So vegetative morphology can really be highly variable depending on light conditions, depending on water, nutrients in the soil. But some of these floral traits just tend to be more stable in general. So this connective dark, generally smooth, versus connective dark, generally wrinkled. I'm going to take that for the second choice, even though some of these leaf characteristics are a little bit more challenging. And this is also the point where if you are unsure, it's always good to key things more than one way. Whenever you come to a couplet that's just really ambiguous, it's good to go down more than one road. And so leaf blade length generally less than 2 times width, filament tube generally less than 3 millimeters wide, root bulblets present at flower, or leaf blade length generally more than 2 times width, filament tube generally 3 to 4 millimeters wide, root bulblets none, which bring us to Primula clevelandii. So I did not find any root bulblets, but that doesn't mean that they weren't there and I missed them. But this is a nice feature here. You can see the filament tube with the millimeter measurement. It's definitely more than 3. So looks like closer to the 3 to 4 range for that filament tube there. So the leaf blade is also sort of a challenging character, knowing exactly where to divide the blade from the petiole when it's a gradual transition like that. But based on this, the character of the filament tube width, we're going to go with Primula clevelandii. So again, this brings us to the description of Primula clevelandii, and we've got some nice photos, so good chance to make sure those look good, but it doesn't end there. There are a few different varieties, so we can go on to these, the key to the varieties. So anther connectives yellow, filament tube without yellow or white spot below each anther, sometimes all yellow, variety clevelandii, or anther connectives maroon to black, filament tube with or without yellow or white spot below each anther. So in this case, there is white in between at the base, but not below the anther itself. So this is the maroon or black connective. So right in here, we have this color. So that will bring us to the second choice. So filament tube without yellow or white spot below each anther would be variety insularis, or filament tube with yellow or white spot below each anther. So again, this is the anther is right here. And so right below that, we have a big dark maroon anther connective with no spot. So that would bring us to Primula clevelandii variety insularis. So again, a good opportunity to check the photos, check out the description, and then look at the distribution. So in this case, it's a Channel Island plant, occurs in the Channel Islands and the adjacent coast nearby. So thanks again for the folks at Tilden for providing us some of this material. I also want to point out that there's some great Primula Jepson videos. So I encourage you to check those out and we can take questions.